with us today, Simona Antonios. Simona Antonios is a PhD student at the INRAE, Toulouse, France, and she has a strong curiosity in developing and understanding quantitative genetics approaches. She's particularly involved in evaluation, design, and optimization of breeding programs. More precisely, she's interested in finding new ways of considering genetic diversity in native sheep breeds for sustainable future breeding programs. We thank you for being with us today, Simona, and the floor is yours.
Thank you so much for the presentation, and thank you everyone for being here to assist to my presentation. So just to confirm with you, you can see clearly my slides?
Yes, we can.
Okay. So as you said, I'm a PhD student. I'm in my second year. So I will present for you my work on my first year, and it's about the partition of the genetic trend of French dairy sheep in Mendelian sampling and long-term contributions. So a little brief about me. I'm of Lebanese nationality. I had my agriculture engineer degree in 2019 at the Lebanese University Faculty of Agronomy and Veterinary Medicine. So during these years also I did my master degree project on the estimation of inbreeding depression for semen trait in dairy sheep at INRAE Toulouse at Genfis lab, at the same lab that I'm doing actually my PhD. So this work ends with a paper that I published in Journal of Dairy Science in 2021. It's about genomic and pedigree estimation in inbreeding depression for semen trait in the Basco-Bernese dairy sheep breeds. And thereafter, I started my PhD in 2021. So right now I am in my second year of my PhD it is also in Genfis lab at INRAE. So right now for the plan, I will start with a small introduction about the subject. Thereafter, I will show you the materials and methods that I used, going to a small example to explain about those materials and methods. Thereafter, to the real data in dairy sheep breeds, to continue with the results that we obtained, and then to conclude with small conclusions. So selection is based on estimated breeding values. However, Mendelian sampling is important in creating genetic progress because Mendelian sampling is the unique portion of the genetic variation that the individual brings in the population. It represents the deviation arising from recombination and segregation. So the breeding value of an individual can be decomposed as a sum of Mendelian sampling of all its ancestors. Thus, the genetic progress can be understood as the selection of positive ancestors Mendelian sampling to their descendants and also the candidate own Mendelian sampling. Thereafter, the long-term genetic contribution of an individual is its proportional contribution to the long-term genetic background of the population. So the genetic contribution, RIG, is defined as the expected fraction from ancestor I, born at a time T, transmitted to the descendant G, born at a later time T2. So the long-term genetic contribution of an individual depends on the genetic superiority for selection of the individual. For example, if we are selecting based on the estimated breeding value, so the individual with the highest estimated breeding value intra-generation will be selected to have more offspring. So thereafter, it will increase their long-term genetic contribution. So the objective of this work was to partition the genetic trend in Mendelian sampling and in long-term genetic contribution. So the study of Mendelian sampling trends by groups and categories of animals. In our case, it will be the sex and selection pathway that I will represent them later on. This allows understanding of selection decisions. So the study of long-term genetic contribution allows to understand the gene flow across generation and by groups and categories of animals. So we analyze genetic gain for milk yield in French dairy sheep. Right now, I will go to the material and methods. As we know, the breeding value of an individual is equal to the average of its parental breeding value, which is known as the parental average, plus its Mendelian sampling term. This Mendelian sampling captured the originality of the animal. So in matrix notation, we can write the vector of the breeding value as a linear combination between the matrix T and the vector of Mendelian sampling. This matrix T is a lower triangular matrix that shows the flow of genes through the pedigree. And it shows also the relatedness between the individual and his ancestors. So this equation also holds for unbiased predictors as BLOP, such as follow, where it is the estimated breeding value, the vector of the estimated breeding value is equal to the linear combination between the matrix T and the vector of the estimated Mendelian sampling. And from this equation, we can deduct the estimated Mendelian sampling such as follow. So by using this equation, Garcia-Cortez proposed a set of K partitions where their sum is equal to the matrix I, where these partitions are dependent on the category of interest. What is P? P is a diagonal matrix that shows and selects the corresponding columns of T, and also it contains zeros or ones at the corresponding column. So from this equation and from this idea, we can write the estimated breeding value such as follow, and by replacing the Mendelian sampling term by its value, we can have the estimated breeding value that is partitioned into K group, where the UK is the part of the breeding value of the category K. Starting from this equation, each breeding value can be decomposed into a sum I of independent terms or contributions that involve the breeding value of founders and the Mendelian sampling of non-founders. So the coefficient TIG is the genetic contribution of the ancestor G to the individual I. Williams defined it by RGI is the long-term genetic contribution of the ancestor G to the individual I. Right now, I will go forward to the example with the 7 individual pedigree. As we can see here, this is the example of the 7 individual pedigree, where the blue represents the males and the pink represents the females. So here we calculated the breeding value of each individual in terms of Mendelian sampling by using this equation. As we know, 1 and 2, we don't know their parents, so they are considered as the founders. So the breeding value of the individual 1 is equal to its Mendelian sampling, and the breeding value of individual 2 is equal to its Mendelian sampling, because the parent average is equal 0. And we continue with the other individuals where we have their parents that are known. For example, for individual 4, where it is the progeny of individual 1 and 2. So we can write its breeding value is equal to the half of the breeding value that comes from the parent 1 and half of the breeding value that comes from parent 2 and its Mendelian sampling such as follow, where we replace each breeding value by its value and we obtain this equation. And we continued with each individual to arrive to the last individual. Note that I represented each individual by a color. Like this, we can differentiate the part of the Mendelian sampling that were coming from each ancestor that were different. So I will explain more about this. So I will use the equation that I described before, and I will take the individual 5 as an example. So here we have the individual 5 that its breeding value is equal to the sum of T and the Mendelian sampling from the ancestor G. The T matrix is shown here, where I is the lines and G, the ancestors, are the columns. So for example, for individual 5, we have half from the Mendelian sampling from the parent 1 plus quarter from the Mendelian sampling that comes from the parent 2, that is here, plus half from the Mendelian sampling that comes from the ancestor 3, which is here, and half of the Mendelian sampling that comes from the ancestor 4 plus its Mendelian sampling. And so on, we can explain it for all individuals in the same way. So right now, I will go with the real data in dairy sheep breeds. About the 4 breeds, I'm working with 4 French dairy sheep breeds. The first breed that I'm working on is located in the Roquefort area. This is France, and this is the Roquefort area that groups the breed that's called Lacon. Lacon breed produce the famous cheese, which is Roquefort. And the other 3 breeds are the Western Pyrenean breed that comes from the Western Pyrenean area. They are Basco-Bernaise, Manège Tête Noire, and Manège Tête Rousse. They are famous also with the production of the cheese that is also Irati. I will explain to you a little bit about the selection scheme of the ovine dairy sheep breeds. As we know, we have the nucleus here. In the nucleus, the breeders are involved in selection. So here, it occurs the official milk recording. All the males are selected on ancestry. So the best lambs are bought by the artificial insemination center, where the testing of young male occur. And thereafter, the males that were selected based on this testing are sent to the artificial insemination center, where they are tested again, and then the males that were selected, they will send their artificial insemination with fresh semen, because with ovine breeds we use the fresh semen, to the nucleus as breeding males. And during this process, we have the sale of breeding animals from the 3 phases to the out of selection nucleus, where it happens the simplified milk recording. So we can see and we can say that this selection scheme starts from the official milk recording and artificial insemination utilization. Thereafter, in this part, we will have artificial insemination and natural mating sires selection. Thereafter, to the final step, that is the dissemination in the commercial flock. So for our data, animals were born between 1985 and 2016. 2016 because it's before the genomic selection starts. And on 1985, it's the year where all breeds have their pedigree and their performance recording started. Those are my 4 breeds that we worked on. Note that the Lacon is the largest French dairy sheep breed. As shown here by the number of animals, they are separated into males and females. The BB is Basco-Bernaise, MTN Manège Tête Rousse, MTR Manège Tête Noire, and Lacon. So those are the 3 Pyrenean breeds, and this is the Roquefort breed. So EBVs for milk yield based on 2021 official national genetic evaluation single step GPLOP, and the pedigree were used to compute the analysis. So the first part was about the study of Mendelian sampling trend by category of animals allow an understanding of selection decision. So based on this equation, the partition of the estimated breeding value based on the category of interest, as I said before, we will work on the sex. In the sex, we have the two sexes, males and females, so K is equal to. And for the selection pathway, we have 5 categories that are dam of males, dam of females, artificial insemination males, discarded artificial insemination males, and natural mating males. Why we have discarded artificial insemination males? Because after progeny testing, those discarded artificial insemination males weren't good enough after progeny testing to contribute their genes, so they stopped contributing their genes thereafter. So here we can partition the estimated breeding value into 5 categories, but here we can partition it into 2 categories. We did this analysis with the alpha part R package that was proposed by Hofstetter and her co-authors, which implement the method of Garcia-Cortez in his paper. In the second part, we wanted to analyze long-term genetic contribution. Why? Because analyzing long-term genetic contribution of ancestors to descendants allow understanding the gene flow across generations and by categories of animals. So for this study, we decided to take the Basco Bernese to represent the Western Pyrenean breeds versus the Lacan breeds. So for each individual eye, it had its long-term genetic contributions, Ri, where Ri is equal to the sum of all the contributions from the individual eye or the ancestor eye to the individuals that were born in the same cohort, where N is the sum of all individuals that were born in the defined cohort. So as I said before, Ri is the same as TGI. So it's the long-term genetic contribution from the ancestor I to the individual G. It is measured as a ratio, so their value varies between 0 to 1. 0, no contribution, 1, it's 100% contribution. So to determine this long-term genetic contribution, we decided to do a pseudo-generational scales for 4 years. So we grouped the individual into 4 years interval. And as I said, the individual were born between 1985 and 2016. So we will have the first generation that groups all the individuals that were born between 1985 to 1988. This is called G0. Then to the last generation that groups individuals that were born between 2013 and 2016, this is G7. So in total, there are 8 cohorts. So right now, I will go to the results of the real data. After using the alpha part for analyzing, the results that obtained were shown here. So here I will show you the decomposition of genetic trend for milk yield in Mendelian sampling term by sex. So this graphic shows the total genetic trend for milk yield and partition into contributions due to Mendelian sampling by sex, where the red line represents females and blue line represents the males, and the black line is the overall genetic trend. So we can see from this graphics that the female contributions for the 3 Pyrenean breeds that are Basco Bernese, Manège Tête Noire, and Manège Tête Rousse are as important as male contribution for the final genetic gain. So we can see that the Mendelian sampling contribution cumulated around 50% for females and males in 2016 for the 3 Pyrenean breeds. But for the Lacan, we can find that the females' contributions are larger than males' contributions. So we can see here that the females' contributions is around 69% of the overall genetic trend in 2016, and the males is around 31% of the overall genetic trend in 2016. Next, we did the decomposition of a genetic trend for milk yield in Mendelian sampling term, but here by categories. So as defined before, the categories were artificial insemination males presented in black, dam of males presented in green, dam of females represented in purple, and natural mating males represented in blue, and discarded artificial insemination males represented in red. So we can see that for the Pyreneans breed, the contribution of artificial insemination males was followed by the dam of males and then by the dam of females categories for the three Pyreneans breeds. But for the Lacan breed, we can see that dams are as important as artificial insemination males in driving change in milk yield. And as expected, we can see that the discarded artificial insemination males and natural mating males did not contribute to the genetic trend in Mendelian sampling term. So here, even if artificial insemination males are more heavily selected, half of their genes come from their dams, and more precisely, dam of males, whose positive Mendelian sampling are eventually selected and spread through the population. And also, the dam of females can be the maternal grandmother of artificial insemination males. Thereafter, we did after processing the output of alpha part, the contribution in terms of Mendelian sampling of year T in category G was obtained as the difference between yearly average. For example, here the Mendelian sampling gain for category G at the year T is equal to the difference between the yearly average at the year T for the category G minus the average that was obtained for the category G at the time T minus 1. So I can give you an example, for example, for artificial insemination male. Before, this graphic is the average contribution due to Mendelian sampling for milk yield by year of birth of the categories dam of males, dam of females, and artificial insemination males. So how we did this calculus? Here in 2001, for the artificial insemination male, how we did it? So the average contribution to Mendelian sampling in this year is equal to the difference between the 2001 point in the trend of Mendelian sampling for artificial insemination male in the slide before, here, minus the point that is in 2000. So like this, we can obtain the all values for each year from each category. So what we can see here is that the Mendelian sampling for the 4 breeds is above 0, but we can find more variation or more dispersion for the artificial insemination male than for the other categories, and this is due to the lower number of artificial insemination male at each year compared to the dam of males or dam of females. And also, we can see that between breeds, we can find that the Lacan has more dispersion, less dispersion than the other Pyreneans breed, and this is due to the lower number of artificial insemination males in the Pyreneans breed. And here we will continue with the other categories. So in this graph, it shows the average Mendelian sampling in terms of discarded artificial insemination males and natural mating males. We can find that The average Mendelian sampling varied between -1 and 1 for the discarded artificial insemination males. And that is because, as I said, they are not good enough after progeny testing, so they stop contributing their genes. And for the natural mating males, we can find that the average contribution due to Mendelian sampling is around 0. And this is because after initial selection based on parent average, natural mating males were not further selected, so no contribution was obtained through their progeny to the selection response. So for the next part, it was about the analyzing of the genetic contribution for Basco-Bernese and Lacan. We can see here that the males in both breeds had higher average individual genetic contribution to the population than the females in both breeds. And this is due to the larger number of offspring per sire. And when we compare between breeds, we can see that higher genetic contributions was found in the Basco-Bernese breed than in the Lacan breed. And this is because that at each generation, we have 10 times lower number, the number of animals in Basco-Bernese breed are 10 times lower than the number of animals in Lacan's breed. And I gave an example in the last generation, G7, we have around 21,000 animals for the Basco-Bernese breed versus 185,000 animals for the Lacan breed. And note that in this graphic, this box plot is done with the log 10 of the contributions of males and females. So thereafter, we wanted to see and to analyze the differences between Lacan breed and the Western Pyrenean breed. So in this part, we took the Basco-Bernese to represent the Pyreneans breed. Those graphics are extracted from the first graphic that I showed. Here, as I said, we have the Mendelian sampling contributions cumulated around 50% for females and males in 2016. Thus, for the Lacan, we have the females' Mendelian sampling contributions were around 69% versus 31% for the males' Mendelian sampling contributions of the genetic trend in 2016. So to see these differences, we decided to do a table to show what's different between both breeds. So in the slide before, we see that there's a difference between females and males contributions because females' Mendelian sampling contributions were around 69% from the genetic trend, but the males' Mendelian sampling contributions was around 31%, which is higher than the difference between females and males' Mendelian sampling contribution for Basco-Bernese because there was around 50%. We can add to this that we have higher generation interval in Basco-Bernese breed because we have a generation interval of 3 years in Lacan versus 4 years in Basco-Bernese breed. Additionally, we have higher insemination, artificial insemination rate in the nucleus for the Lacan breed than for the Basco-Bernese breed because we have around 85% of the artificial insemination rate in the nucleus versus 55% in the Basco-Bernese breed. So we can conclude here that we have larger family size. So we decided to see the family size for the artificial insemination male. We thought that we have for each artificial insemination male around 110 progeny that are females. And we have, this is for the Lacan breed. For the Basco-Bernese breed, it was around 90 female progeny for each artificial insemination male. So here we can conclude that we have higher accuracy and the selection originality will be more efficient in Lacan breed than in the Basco-Bernese breed. Later on, with the analysis that we did, we remarked that the selection on Mendelian sampling was more important in Lacan breed than in the Basco-Bernese breed. Thus, the selection based on parent average was more important in Basco-Bernese breed than in Lacan breed. What's the importance of this? Because the difference of the relative importance of the Mendelian sampling and parent average between breeds across generation could be and could reflect the difference between selection strategies, because the selection based on Mendelian sampling relies on within family selection, and the selection on parent average relies across family selection. So as we can see here, because we have higher selection on Mendelian sampling in Lacan, we can say it is possible to have higher within family selection for the Lacan versus to have higher across family selection for the Basco-Bernese breed. So we can say a larger family size in Lacan than in Basco-Bernese is in agreement with a greater importance of within family selection in Lacan, which could be because it is possible to exercise higher selection intensity within family when family are larger, as is in the case of Lacan. So right now we will go to the conclusions. The first conclusion was that the females contribute largely to the genetic trend by being mother or grandmother of an artificial insemination males. Secondly, the fine partitioning in Mendelian sampling, as we saw in the graphics, demonstrates that females are the main reservoir of genetic variability. But we don't have to forget that the artificial insemination males are crucially important because they allow the spread of the best genetics in the population and genetically connect the herds. And finally, the Mendelian sampling term captures the originality of the individual with respect to its parent and has a role in creating genetic progress. Thank you for your attention.
Thank you very much. Her presentation was absolutely amazing. Does anyone has questions? If anyone has questions, you can raise your hands and turn on your cameras and microphones and ask Simona. While we're waiting for, oh, okay, so Flavio has a question. You can go, Flavio.
Okay. Simona, great presentation. I missed the very beginning of your presentation. I was rushing to get home. So I have a question for you. Based on the time frame of your study, you removed the more recent data that was subject to genome selection. Was that the case?
It was prior to the genomic selection because
okay. So it was prior. So everything that you showed here, the partitioning of the variants, the Mendelian variants, was prior to genome selection.
Yeah, exactly.
Yeah. So I have a question for you. What are your expectations if you look at the same data, the same statistics, the same partitioning? If you do it after genome selection, what do you expect are going to see? The differences?
After genomic selection, because after genomic selection, even we have different selection scheme decisions. Because, for example, before genomic selection, we had the discarded artificial insemination males that were presented. But after the genomic selection, the selection is not based as before on only the progeny testing. They will test the youngest animal directly, and they will start doing the scheme and the breeding. So
How about the fact that before with just the blood theory, right? In fact, Mendelian sampling was a completely random process that we could not really kind of use it, right? Only when the animal had their own performance, then we could deviate that performance from the form of the parents and have assessment of the Mendelian sampling. But now with genome selection, based on the marker information, you can select based on, in fact, we do that, we select based on the Mendelian sampling, even when the animal don't have the phenotype. So that idea that Mendelian samples are going to recover 50% of genetic variance each generation, right, is not going to happen anymore. So in that sense that I'm asking, if you thought what would be the results, how the contributions that you look, for instance,
Do you mean
for the genetic trends, now the maternal side contribute a lot for the trends and so on.
Do you mean the contributions of the different selection pathways?
Yeah, yes, in through the Mendelian sampling.
Yeah, I think that by using the Mendelian sampling and the genomic selection thereafter, I guess that the females will always have this, how to say, this contribution. Because before we only had it, we only had the information based on the artificial insemination males, and this is why we can see that even for the, I will show you here, even for the Pyrenees breed, because Lacan is much more elaborated with her selection scheme. That's why we can see and we can spot the difference. We can see here for the Lacan breed that the contributions is as important in the same case. And I guess for the future, if they will follow this, how to say, this strategy, they will all arrive to this contribution for the different selection pathway. For the dam of males, dam of females, and artificial insemination males will be the same importance, plus or minus. I don't know if I answered your question, but
yeah, I think it's difficult to answer. It's just I think we need to look at this as more years of genome selection accumulates, right?
Yeah.
Because it's changing the structure of the population, as you mentioned.
Yes,
the
exactly.
It's changed, but also this fact that now you can really, before the animal has any own record, we can have assessment of the Mendelian sampling of the animal, right? With based on the marker effects. And I have my last question. I don't like to take all the questions, but you in the study you assume a generation of 4 years, right?
Yeah.
For all breeds. But later on in your talk, you show that the Lacombe has a generation interval that's 3 years, not 4.
Yes.
So
because
would that affect your results because you assumed a longer generation interval for Lacombe when Lacombe had a shorter generation interval?
Because it's not a real generation. Well, in the sheep, the generation is around 4, but in this study we took it as a pseudo generational interval. So we took it as a group of individuals. So it's not a real generation; it's a group. So we called it group 0, group 1, group 2, group 3 till group 7. So it's only a generalized thing that we decided to have groups of 4 years for each breed to have the same to have the same analysis for both of them.
Yeah, but would not be more realistic if you used age interval for each breed if they if they were different, even though you create a pseudo based on years, right? But it will be closer to the actual generation interval of each breed. I don't know.
because the thing that we wanted to see is the contribution for the individual to the cohort. So to see for the individuals that were born between this year and this year, what was their contribution to the individuals that were born between those years. So like this, to have an accurate analysis between both of them, because we didn't want to have different groups of animals that were formed with different years. That's why we said, okay, we will take this pseudo-generational scale. So it's, if we...
I understand, but I could argue that for Lacombe, the contribution would be faster.
Yeah.
Other breeds, so in that you didn't take into account. But that's just my opinion. But great presentation, Simona. Thank you for doing this.
Thank you so much.
Thank you, Flavio. Does anyone has any questions? Anyone else? Okay, so I just have like...
Maybe there is no other question. I just going to ask Simona, did you publish all these studies?
Right now, it's in the revision with the Journal of Daily Science.
Okay. Okay, I like to keep the eye on the
I will keep you posted. When it will be published, I will send you an email.
Okay. Thanks, Simona.
Welcome.
Okay, so anyone has any more questions for Simona? So Simona, just have like more for curiosity question. When papers related to Mendelian sampling, like yours, start to be published, like when people start to worry about this part that we couldn't measure that much?
Can you repeat, please?
Yes, of course. So when papers like studies related to Mendelian sampling start to be published, like similar to yours or something close to that?
Well, the things is that according to according to this study, it was the first work that was within the country and within because the study that was done before was with the introduction of foreign animals, but this study is the first one of its type because it's in the breed, in the closed breed, so pure breed. But the studies for Mendelian sampling that started with Garcia Cortez, and I put the references here. Started with Garcia Cortez in 2008. And also there was another paper, I have to remember about it. Also, it was about the Mendelian. I can.
No, it's okay. It's just more of a curiosity.
Yeah, because I cited them in here next to me, so I can tell you the names of the papers.
Yeah, no, it's okay. So Flávio has another question.
In fact, is a comment on this. Okay. I think Simona is aware, but there is a very nice paper by Daniel Sorensen and Brian Kenny. I think was published in 1996 or 1994. The title is Genetic Properties of Mixed Model Equations. And in that study, they showed already, they showed that any breeding value of any animal can be shown as a function of Mendelian sampling of the ancestors until you go to the base generation, right? So exactly this decomposition that Simone is showing here. But they show that with another objective, to show that if you have a complete relationship matrix that goes to the base generation, then this relationship matrix accounts for selection, because it accounts for all the flow of the genes. But how to show this is that any animal, even selected in the current generation, nowadays, for instance, the EPV can be expressed as a function of Mendelian sampling terms that goes back to the base generation. And so that's it's kind of how they show that not affected by selection. But of course, the pedigree needs to be complete. Right? So, and just to say that that time, that this idea of the composition in terms of Mendelian sampling was already known. But with new applications now that like Simona is presenting. But that paper is very worthwhile to read. I think every single student that is using mixed model equations should read that paper. And of course, nowadays, again, you have to think it's different, because that's in the base that we could not select based on the Mendelian sampling, right? It was a completely random effect. But now with the markers, we can have assessment, direct assessment of the Mendelian sampling. So it is a little bit different now, the selection bias problem.
So, please, can you repeat the name of the paper?
It's genetic properties of the mixed model equations.
And
by Sorensen and Kennedy. It's published in one of the proceedings of the quantitative genetics meeting. I think it was the second one. Oh, if you like, Simona, I can send you the reference.
Yes, please.
Okay, very, very good. Thank you, Flavio, for the reading paper.
Yeah, thank you.
Does anyone has any more questions? Okay, so I think if no one has any more questions, we'd like to thank you again, Simona, for your presentation. It was absolutely amazing. Thank you for your time and well done job.
Thank you so much for this opportunity also. And thank you everyone for being here to hear my presentation.
Okay, so we'd like to thank everyone that joined us today, and I will soon see you next Friday.